Hello, my name is Kayla Redmond, and today I'll be presenting my REU project. The title of my REU project is Investigate the Diversity of Gram-Negative Bacterial Fatty Acid Transport Proteins, FATs, using protein modeling. Introduction. Previous research supported that gram-negative bacterial proteins are involved in the uptake and assimilation of exogenous fatty acids. The multiple homologs of these gram-negative proteins are presumed to be responsible for the expanded ability of some bacteria to perform fatty acid uptake and assimilation. Several polyunsaturated fatty acid PUFAs have had an important impact on the gram-negative bacterial physiology and behavior. Such impacts include the changes in bacterial membrane phospholipid structures, permeabilities, phenotypes associated with virulence, and consequent stress responses that may impact their survival and persistence. Our research proposal. With the discovery supported in previous research, we believe that protein modeling and bioinformatic assessment of the machinery involved in fatty acid uptake, such as the fat L's and fat D's, could reveal key differences in binding characteristics among gram-negative bacteria. Identifying potential structural/functional differences could help explain how gram-negative bacteria, pathogenic and non-pathogenic, have evolved to scavenge specific exogenous fatty acids for their survival and persistence in specific environments. So here is a diagram of how this bacteria utilizes its fatty acids. So the fatty acids, in this case E. coli, comes through your fat L and it ventures through your fat L's until it gets to the fat D's, and fat D's make this acetyl-CoA. Approach. Our initial approach was to examine both fat L's, which are long-chain fatty acid transporters, and fat D's, inner membrane protein that creates acetyl-CoA. However, the data for fat L's was so extensive that we could not proceed to the fat D's analysis. With the assistance of a phylogenetic tree of fat L's, we were able to use the locus tags to identify all long-chain fatty acid transporters, amino acid sequences through NCBI and IMGM. So here is the phylogenetic tree that I just mentioned that has over 120 different fat L's. And you can see they are grouped respectively to their organisms. Switch model. With the locus tags/identifiers of the fat L's, we were able to gather the amino acid sequence to construct the fat L homologs. Switch model, a fully automatic protein structure server, was used to model each protein. With the switch model tool, we were able to build over 120 3D protein models using the 1T16.1.A template, a crystal structure of the bacterial fatty acid transporter fat L from E. coli. So this is basically how all of our fat L's look because we built them off of a E. coli template. What's next? So after we modeled these 3D structures, which were 120 fatty acid structures, we wanted to look specifically at the known conserved amino acid features, which are amino acids that were predicted to participate in recognizing and transporting fatty acids in each group of the bacteria. So we used clustol. Clustol omega was used to align 11 different groups of fat L's from the phylogenetic tree. These alignments showed high similarity, as we expected, within each group, but they were difficult to interpret with regards to fatty acid binding characteristics. Alignments. After aligning the fat L's with their respective groups, we searched for well-defined amino acids that show importance in the binding of fat L's from previous studies. These amino acids include F3, R15, A21, R32, N33, Q33, P34, and so many more. These are conserved amino acids that compromise important protein domains such as low affinity pockets, high affinity pockets, N-terminal hatches, kinks, conveyor belts, and etc. And here is a picture of the high affinity pockets, R157, L200, F235. So these are just some of the amino acids that we were looking for after we align the protein, the fatty acids. And this is just a picture of how our alignments look. So this is the non-gamma, which is the top section, because one or two sections had so many fat L's in them, we had to break them down piece by piece. Ones that are closely related to each other, we put those together in the same alignment. So as you can see right here where my cursor is where we started off. This is where the fat L's start, their amino acid sequence start. So we had to start right here and subtract that number away from 60, and that's how we'll find if it's a L200 or a G143. So these lines that has the brackets around them, those are the amino acids that we're looking for that was found in previous studies. And sometimes, as you can see, E319, we have a question mark by that because it was such different bacteria, even though they belong to the same group. Some of the amino acid sequences was shifted down, so it was kind of hard trying to keep track of what number you're actually at. Even though all of them had E, but they might not have been in the 319 position, we just put a question mark beside that because of the shift. And we came to a roadblock. Although many of the fat L's showcase the important amino acid sequences or the amino acids that we were looking for, we did not have enough information to conclude that they were an essential part of the binding motifs. So we took a detour. Even though we could not identify specific fatty acid binding characteristics among bacterial clusters, we improved our approach by using the E. coli fat L to perform alignments with each cluster. So we went back to cluster omega. So when we went back to cluster omega, we chose a few of the fat L's from the different 11 groups and we went back and realigned those against the B2344 E. coli fat L. And this is just an example of how it looked. This allowed us to illustrate how each cluster within the phylogenetic tree has its own unique variation within the fat L sequence. Importantly, these amino acid regions are located within the most important domains controlling fatty acid transport. So when we went through and looked at these alignments, and we see, for example, that this is cut off on both of the B2344 and the XAO8, but we want to know what did this signify right here in this area, and we'll talk about that next. And we use PyMo. So with PyMo, which is a software capable of 3D molecular visualization system, we were able to upload the PDB files that we downloaded from Swish Model after we modeled those to view and align the fat L's against one another. The ability to show these 3D models allow us to visualize the different places fatty acids may enter and interact with the fat L's. So this is just one of the one of the many alignments that we did. As you can see that they are color coded, so you can notice the difference, which is explained in the next slide. So this is a crystallized structure of a fatty L. You have your loop 3 and your loop 4, and then you have your N-terminus hatch right here and your kinks. These are what the amino acid that we were looking for in our alignments. And then you see you have fatty acids and how they come in. So the importance of this project was to model these fat L's so we can see where and how does these fat L's come in and which amino acids do they bind to when they're coming to these fat L's. And because fatty acids have such a diverse shape because of their carbons and they can curve their shapes, it was important to see how these loops and the structure of the fat L interact to see which kinds of fatty acids could enter. So this is different fat L's. So the green is E. coli, and these are two other fat L's, X808 and GA031244. And you can see that they are kind of similar, but right in this area where this yellow is showing on both of these figures is where I was saying on the previous slide that our alignment showed that they had extra amino acids in a region where either the other two did not have that excess region of amino acids. So maybe we discovered that because it has this difference, this may be a key for why it uptakes different fatty acids than maybe this structure or this structure. So it's very interesting. And these are pictures of fatty acids, including linoleic acid. So you can get a general idea of how these fatty acids look and why it is so important to know the structure of the fat L's. Because if you have a fat L that is not big enough or doesn't have the structure to allow a fatty acid of this size, which is a certain amount of carbons, then it would not be able to uptake and assimilate. So it's important to know that structure of that fatty acid so you can see how and which fatty acids can be uptaken. Significance. So overall, elucidating the similarities / differences in fatty acid structures of gram-negative bacterial proteins will, one, help us discover which fatty acids can be acquired based on protein structure. Two, predict whether these fatty acids are ecologically adaptive and could be important for bacterial survival and persistence. And three, allow us to better understand how fatty acids are recognized and transported through the gram-negative bacterial outer membrane. And overall, we want to know, why does gram-negative bacterial proteins are so important? So a variety of gram-negative bacteria possesses fat L proteins. And these bacteria are important research because of their effects on living organisms in our environment. So the Vibrio species are known to have a negative effect on human health through the exposure of them in aquatic environments. This goes as far as eating seafood that you from fresh water. These Vibrio species can be on seafood and water, anything that has been around an aquatic environment. Then you have your Shigella species, which are found in warm and aquatic environments, and they can cause soft tissue, ear, or abdominal tract infections. Then we have our Photobacterium species, also found in aquatic environments, which causes diseases in fish, which can indirectly impact human health and the economy. Because if they have been adapted into the fish that are grown in fish farms, then you cannot sell those products to the human race because that's dangerous. Then we have our monas species, which are found in warm aquatic environments. This provides a wide range of intestinal and extraintestinal diseases within the humans. Future research. Future research can be easily extended with the abundance of data generated in this project. With better understanding of bioinformatics and computer science, it is feasible to perform ligand binding simulations using each fatty acid in the model fat L's, such as AutoDock. I wish I knew how to work AutoDock because they have been very important into our project. But like I said, more bioinformatics and computer science is well needed for this project. Also, it'll be wonderful to explore the fat Ds and compare their structures and role to the fat Ls. The fat Ls had over more than 120 species, so the fat Ds may be 120 or more because it was such great information to use. Acknowledgements. I would like to acknowledge my mentor, Dr. David Gills, for his endless support. I would like to acknowledge the NSF REU Award number 1852042, and I would like to acknowledge Dr. Hung in the iComp Bio REU program at the University of Tennessee at Chattanooga for this wonderful experience. And here are a list of my references. Thank you.